So, good morning everyone. So, in our general histology lecture series, we are studying about like basic tissues of the body, right? And then we will move to the systemic histology. Now, as far as like basic tissues of the bodies are concerned, okay, there are, it must be taught, right? There are 4 basic tissues in the human body. What are they? Yeah, yeah. Mano, can you stand up, please, and say it loudly? 4 basic tissues of human body. Huh. Yes. So, epithelium, connective tissue, muscle, and nervous tissue, right? These are the 4 basic tissues of the body, and out of this, today we are going to see the histology of muscle. Now, these are basic, so they are having some special properties, okay, which distinguish these tissues from each other. So, so far we have studied in the previous lectures about epithelial tissue and about connective tissue. What do you, what do you think, what makes muscle separate from these 2 tissues? Because see, muscle is already taught to you in general anatomy, right? So, what do you know about muscle tissue? What is special about muscle? Yes, correct. It is having ability to contract. And what happens because of that? Locomotion, movement, okay, our day to day activities. So, that is all that happens because of this muscle and its property, right. So, this is one of the, we have seen already that muscle tissue is one of the 4 basic tissues of the human body, and it has the special properties, ability to contract, right. So, what happens in contract actually? It shortens in its length. When it relaxes, it comes to its original length. Now, this contraction, this is accomplished by a special cell, and that is called myocyte. So, cell of the muscle is called myocyte, and these cells are held together by connective tissue, that we know. Now, these actually one myocyte forms a muscle fiber, that we have to understand. When we are going to discuss further, we need to remember this. So, myocyte is also called a muscle fiber, and basically these are elongated spindle shaped cells. These cells, these myocytes, are derived from mesoderm. In the days to come, when we will discuss further about embryology, then we will teach you about this. Now, here what happens now, there are special terminologies. In like we have studied the structure of the cell, okay. So, cell is having cell membrane, right. So, cell membrane, or that is also called plasma membrane. Now, plasma membrane in case of myocyte is called sarcolemma. Then cytoplasm, here it is called sarcoplasm. Similarly, endoplasmic reticulum is called sarcoplasmic reticulum, and this sarcosome is mitochondria, right. These are the different terminologies we will come across as we move further in this lecture. Now, we divide this into different types. In general anatomy also we have learnt about it, right. So, histologically, basically today what we are going to see is, we are going to divide the muscle tissue and study them in 3 types: skeletal muscle, cardiac muscle, and smooth muscle. Now, before I move further, this basic points are already taught and touched in our general anatomy lecture. So, Parth is there, no, today? Parth, stand up beta. So, what do you know about this? Skeletal, cardiac, and smooth muscle. In general anatomy, what we have learnt? Hmm? Hmm? Yeah. Yes. So, very good. So, he has explained this entire slide, as you can see. So, there are various ways how we can classify the muscle as a tissue. So, skeletal, cardiac, and smooth. And the skeletal muscle and also cardiac muscle shows striations, so striated, and there are non-striated. So we can divide the muscle tissue into striated and non-striated, and then there are muscles with which we do our voluntary actions. So these are we can divide, we can label them as a voluntary, and there are certain muscles which are not under voluntary control. So they are involuntary. So this is how we classify the muscle. Now let us come to the skeletal muscle first. Okay, and as you know, skeletal muscle is also called as striated muscle, and why? Because it has striations on it. So we will see how these striations are produced. Okay, and then whatever actions we are performing with the help of the skeletal muscles are under voluntary control. So these skeletal muscles are also called as voluntary muscles, and yeah, these muscles are responsible for our locomotion, our movement, which helps us in our all day to day activities. Now there is a special terminology is visceral striated muscles. So like these striated muscles, they are like present in some viscera. Okay, so like some organs. So basically microscopically, if you see, their structure is identical to the skeletal muscles, but like they can be, they are present in the tongue, they are present in the pharynx, peripheral part of the diaphragm, and even in the upper 1/3 part of the esophagus. So instead of locomotion, here they are helping us in speech, in our breathing, in our chewing action, okay, swallowing and coughing. So this is like we need to know about the visceral striated muscle. I hope you are getting me. Any doubt so far? Yeah. Now, if we take a small part of the skeletal muscle and take a sections of it and observe it under microscope, we can do it two ways. We can take a longitudinal section and study it, and we can take transverse section and study it. So now this picture, this is the longitudinal section of a skeletal muscle. So now what we see here is, now this is one muscle fiber, this is another muscle fiber. So these muscle fibers or myocytes, they are running parallel to each other. Now what happens now, when this myocyte are being formed, myocytes are derived from myoblast. Now these myoblasts during developmental period, what they do is they fuse with each other. Many myoblasts fuse with each other, they lose their cell membrane, okay, and what happens now, there becomes one single myocyte, but the nuclei of different myocytes are there. So in one skeletal muscle ka fiber or myocyte of a skeletal muscle, we see a long cell with many nuclei. So there are, so myocyte in the skeletal muscle is multinucleated. That one we have to remember, and these nuclei, they are pushed towards the periphery, they are towards the peripheral part, towards the cell membrane of the myocyte. So that, and then now these, the very peculiar feature, you can easily see, you can appreciate these striations. Okay, so these striations are very obvious. So these are because of alternate dark band and light band. So these are the basic features of the skeletal muscle in longitudinal section. So what we see is, we see parallelly running muscle fibers. Muscle fiber basically a single myocyte. Okay, it is a syncytium. There are many myoblasts, they fuse together and form a single myocyte, but they retain their nuclei. So it is a multinucleated cell, and these nuclei are flat, pushed towards the periphery. This, and then they show transverse striations. These striations are because of alternate dark band and light band. Now why this dark band and light bands are produced, we will see in the next slides. Okay, before that, let us have a look on the transverse section, how this skeletal muscle looks under transverse section. So basically what we see is, as we have discussed also, like connective tissue, what is their function is to connect and support, right. So, what connective tissue doing here is, connective tissue is surrounding individual muscle fiber. Then these muscle fibers come together, they form a bunch that is called a fasciculus. So, the same connective tissue surrounding that fasciculus, and then these fasciculi coming together and form one muscle, like you have seen biceps, we are studying about gluteus maximus, whatever muscle it is. So, it is having the same structure, skeletal muscle. It is the muscle is made up of fasciculi, and fasciculi are made up of individual fibers, right. Now, entire muscle is covered by a covering that is called epimysium. So, this one is epimysium. Now, this epimysium, this connective tissue sends septa inside, and this septi, what they do is they cover the muscle fascicles together, and that covering of the connective tissue which is covering the fasciculus is what? Say it loudly. Perimysium. And then again it sends septi inside. This septi covers individual muscle fiber, and that one is endomysium. So, these are the three coverings, endomysium, perimysium and epimysium. Okay. And this is visible when we take transverse section of the skeletal muscle. So, this is the schematic diagram actually. Okay. The actual diagram we will see in the next slides. So, and what happens now, wherever there is a connective tissue and wherever there is a space that is occupied by this blood vessels and even the lymphatics. So, this is what we are going to see in the transverse section. We are going to see the muscle fibers coming together and forming the fascicles. So, this individual muscle fiber, say it loudly with me, is covered by endomysium. Fascicles are covered by perimysium, and entire muscle is covered by epimysium. Any doubt in this? Good. Now, this is the transverse section of the skeletal muscle. As we can see here, see this is a 40x view and this is 100x. So, high. So, now here we can make it out, this is individual myocyte or muscle fiber, this is another one, this is another one. So, here we can see this, actually there are multinucleated cells, right. So, we can see the nuclei which are pushed towards the periphery, and this is the which mysium? Endomysium, right. Now, this coming together and forming one fascicle. So, this is one bundle. So, this is covered by perimysium. So, this is also perimysium, and now many fasciculi coming together, and then they will be covered by epimysium. Now, epimysium is not visible in this particular slide, right. So, here also like we can see this thin endomysium, and then outside this thick will be the perimysium. Now, let us see the further detail about the skeletal muscle and how dark band and light bands are produced, and how contraction takes place. This is we are going to discuss in further slide. Now, what happens now? We have seen, see, the entire muscle covered by what? Epimysium. Now, this is muscle. Now, muscle is made up of many fascicles. These fascicles or bundles are covered by perimysium. Now, fascicle having many fibers, right? Many muscle fibers, okay? And individual muscle fiber is made up of many myofibrils. So, this is one muscle fiber and it is made up of many myofibrils, as you can see in this diagram. Now, this myofibril, now when we see, it is the structural and functional unit of the muscle fiber, myofibril, and these myofibrils are again arranged like if you see like any muscle, in it they are parallel to each other, longitudinally arranged, and this contain many myofibrils, they contain myofilaments. Now, these myofibrils, they are having dark band and white band. Why we are going to discuss in further slide, but as they are arranged one above the other, like you can put your fingers one above the other, like this. Put your fingers. and you can see there is a crease between the uh, two phalanges. So you can just put two creases one above the other. You put like this, as I put. Now see this crease and this crease, like creases one above the other. So there are many myofibrils put one above the other. So just imagine these creases coming one above the other, isn't it? So as they are longitudinally arranged and all these like bands, now just this assume in this case the creases. So they are coming one above the other, continuous, and that is why we can see these bands clearly in case of skeletal muscle. In cardiac muscle, there are these bands, but what happens? No, they are not arranged one above the other like this, because cardiac muscle branch shows branches and then anastomose. So because of this, no, this arrangement is lost, and that's why though there are striations, they are not visible in under microscope when we examine them. So that is the difference, and that's why we see striations in the skeletal muscle. We do not see, if you look very carefully, even we can see them in the cardiac muscle as well. So yeah, this is clear. So we have seen structure. I will repeat again. The muscle made up of fascicles or bundles. Bundles are made up of individual muscle fiber, and muscle fiber is made up of myofibril, and myofibril show this bands. So so there are dark band and light band. So dark band is called A band, light band is called as I band. So further we discuss about this. So now this arrangement is because of the filaments which are there, thick filaments and thin filaments. So myofibril is having these filaments, thick filament is myosin, and thin filaments are actin and even tropomyosin. So what happens now when we under microscope, when this light passes through the dark band, it get diffracted and does not reach the observer again. And because of that, no, we see dark band. That dark band is also called anisotrophic band. A stands for anisotrophic band. While in light band, what happens now, when the light goes to that band, light get diffracted, does not get diffracted, and that's why we see the light there, we see the light band there, and so it is called isotropic band, and that's why the name I band. So A band anisotropic and I band is isotropic band. So now in the center of the I band is a light band. In the center of that we see one dark line, and that is called Z line or Z disc. So that is what, and then in the center of the dark band, that is A band, we see a narrow lighter zone, and that is H band. Okay, and again in the center of the H band we are going to see one line, that is the M line. So all this we are going to see in the diagram. So and there is very important terminology that is called sarcomere. So this sarcomere is basically the functional unit. Actually the actual functional unit is the sarcomere, and what sarcomere is? It is the region between the two Z lines, that is the sarcomere. Let us have a look on this diagram. So before that, just we can see the skeletal muscle fiber, we can divide, we can see there are dark bands and there are light bands. Dark bands are also called as, say it loudly, light bands are also called as, yes. So in the dark band we see a lighter zone, that is called H band, and in the center of the H band we see one line, that is the M line. While in a dark band, in the light band, in the center we see one more line, that is the Z line, and the area between two Z lines is called as sarcomere. So here we can see this is the dark band, this is the light band, dark band, light band. Okay, now in this light band, can you see this line? This is the Z line. Now here this is the dark band, and in the dark band here is the lighter H zone, and now this is Z line, this is the Z line. Now this area, this particular part is called as sarcomere. Clear? Yeah. So, again here we are going to see that again. So, here we can see this. So, this, can you appreciate this red line, thicker line? So, this is the thick filament, the myosin filament. Now, in between these thick filaments, can you see these lighter lines? These are the thin filaments, the actin filaments. Okay. Now, when you can see here the area where only thin filaments are present, that particular area is I band, right, or the light band, and where the area where both thin and thick filaments are there, that is the which band? A band or the dark band. And you can see this, what is particular about this area? Only thick lines, only thick filaments, where the area where only thick filaments are there, that is the H zone or H band, right. And you can see there is a, where these actin filaments are connected with each other. See, this is the Z line. So, this Z line and this Z line, this particular part is called sarcomere. So, sarcomere is clear with you, everyone? Any doubt you can ask me, I can explain again in the sarcomere. So, if you see, this is the schematic diagram. Now, here you can see this, see, I band, only thin filaments are there, right. Now, this is the H zone of the dark band, here only thick filaments are there. Now, here this is the, so this is the M line in the center, and then we can see outer edges of the dark band where both the thick and thin filaments are there. This is this clear? Now, how the contraction takes place? For that, there is a theory called as sliding filament theory. So, what happens, these filaments, they slide over each other, and because of that this contraction takes place. Now, we have seen that the muscle fibers are made up of myofibrils, and myofibrils are having sarcomeres, right. Sarcomere is actually the structural and functional unit of the muscle. Now, these are overlapping each other, as if you can see the here. Can you see that this filament, thin filaments are overlapping the thick filament. Now, when this overlapping changes, contraction takes place. So, what happens is, you can see in this, can you appreciate this? So, this are thin filaments are coming closer. So, when this Z, because of that what happens, Z lines are coming closer. So, because of that I band and H band become smaller. Can you appreciate that? Here also see, so I band has reduced, even H band has reduced, there is no change in actual A band. So, that is what happens, and because of the sliding of the thin filaments, so this contraction takes place. Any doubt in this? Shall we move to the next? Yeah. Now, how this takes place? So, basically all this contraction, this is basically dependent on calcium, calcium release. So, this will be taught to you in histology in detail. Okay. But just to make you like what happens now, usually main thick filament is the main filament here is the myosin, the thick filament. It is having this head. Can you appreciate head of this myosin filament? So, this head of the myosin filament is usually occupied by the tropomyosin. Now, when calcium is released, calcium causes the wave of depolarization, and so that what happens, tropomyosin get unbound. So, then it gets bind with the actin, and when it binds with the actin, as you can see in the next diagram, then it is getting bound, and then it moves further, and because of that what is happening is actin is moving, actin filaments is moving, and because of that sliding takes place. Okay, as short as possible I just tried you, but detail of it will be discussed in physiology class. Any doubt in the skeletal muscle? So just to summarize, can anybody summarize? Anyone? Yes, beta. Yeah. Yes. Yes. Hmm. Hmm. Huh? Yes. Myofibrils. Yes. Yes. Myofibrils are myofilaments. Yeah. Correct. Hmm. Endomysium. Right. Yeah. Hmm. Hmm. Yes. Go ahead. Hmm. Yeah. Yes. Yeah. Yeah. I band. Yes. Hmm. Right. Very good. Yeah. 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 Hmm. Correct. Right. Very good. Any doubt? Yeah. So let us come to the cardiac muscle now. So cardiac muscle, as we discussed earlier, it is also having striations, but as I told you, it is relatively difficult to see those striations under microscope because of the branching. So this is involuntary muscle. Cardiac muscle, the involuntary. It is basically you will be taught later. There is SA node, there is AV node, how they generate impulse and how the this contraction is generated. So it is a self-excitatory muscle basically. Now here their nuclei, striking feature is nuclei are not at the periphery, nuclei are in the center. And another point you have to remember is they are branched. So muscle fibers are branched and nuclei in them are present in the center. So as this is the diagram we can see, where we can see that these individual muscle fiber, again they are also running longitudinally, they are parallel to each other, but they branch. So here you can see this branching. Here also suppose take this and you can see this branching, they branch and their nuclei are usually in the center, sometimes single nucleus, sometimes two nuclei also we can see there. That is the main thing. Now these are also striated, but usually they are not that easily, striations are not that easily visible. Now these branched myocytes or branched muscle fibers in the cardiac muscle, they are connected with each other by specialized junctional complex, and that is called as intercalated disc. So you can see here in this diagram. So these are this is where you can make it out. So the adjacent myocytes in this cardiac muscle connected to each other, attached to each other by specialized junction, and that is called as, say it loudly, intercalated disc, right. So here we can see these muscle fibers, they are running parallel with each other, but they are showing branching, okay, and they again anastomos also with each other, and these nuclei are present in the center. Here also there is endomysium, there is perimysium, and now epimysium actually is replaced by a connective tissue covering that is called as epicardium. Instead of epimysium, there is epicardium. As you can see in this diagram, these are the individual myocytes or muscle fibers of the cardiac muscle covered by endomysium. Nucleus is in the center, and this is a fasciculus covered by this perimysium, and then entire muscle is covered by epicardium. So, this is the longitudinal section and this is the transverse section of the cardiac muscle. So, what we have to remember is cardiac muscle, again muscle fibers are running parallel to each other, but the striking feature, differentiating feature from the skeletal muscle is they are branched, they show branches, they again these branches anastomose with each other, and nuclei are where? At the center. In the skeletal muscle, nuclei where? Pushed to the periphery, right? That is the difference. And then these myocytes, adjacent myocytes, are connected to each other by intercalated discs. So, this is what we have seen. So, cardiac muscle having branching fibers, cardiac myocytes. So, now these are also like shows striations, and when we see nucleus, no, we can see a little hollow around the nucleus, that is called perinuclear hollow, not very clearly seen in this diagram, but we can you appreciate this area here. So, this kind of a little bit hollow, little bit empty area around the nucleus. So, perinuclear hollow can be visible. Nucleus is single, nucleus is large, oval in shape and centrally placed. These are the features. And intercalated disc, it is the specialized cell junction, so between the two adjacent myocytes, and it consists of these three structures: fascia adherens, macula adherens and gap junctions. So, with this, these are connected with each other. Yeah. Any doubt in cardiac muscle? Anyone can just summarize the cardiac muscle, please? Aharth, let Aharth do it. Yes, beta, stand up.
The cardiac muscles are the muscles of heart.
Yes.
They are striated, but the main difference between the cardiac muscle and the skeletal muscle is that they are branched and they are involuntary. And in the LS of the cardiac muscle, we see the striations, and the branch fibers are connected with each other with the intercalated disc.
Yes.
Nucleus central.
Nuclear center.
Only mostly one nucleus present for the contrary to the skeletal muscle, which are syncytium.
So, sometimes two also, right?
One
or two nuclei.
And there is a clear area next to the nucleus, which is called the perinuclear hollow.
Perinuclear hollow. You can visible here also. Yeah, here. Yeah, here. Right. Huh?
Yes, and the epimysium is replaced with the epicardium.
Yes.
And the endomysium is highly vascularized connected.
Good. Clear, everyone? Sit. Good. So, let us come to the smooth muscle. Now, the smooth muscles are non-striated. As they are non-striated, that is why they are called smooth. There are no striations, so they are smooth. So, okay, you understood this? Their shape is fusiform. These are now in these, no, there is a single nucleus which is placed in the center. So, with the help of this, with the fusiform shape of the muscle fibers or the muscle cells, the nucleus which is present in the center, we can differentiate them, and they are smooth. Why they are called smooth? Because there are no striations. These are involuntary muscles, and by this neuroendocrine, they are under neuroendocrine control, and that modulates its contraction. And these muscles we can see in the blood vessels, we can see them in the urogenital organs, and even we can see in the skin. Arrector pylorum muscle will be taught later. You want to see? Huh? Yeah. Go. Now, here we can see both the sections. So, this is the longitudinal section and this is the transverse section. So, in the longitudinal section you can make it out that this is the spindle shaped or fusiform shaped cell and nucleus is in the center. And here the transverse section, we can see this again the muscle fibers coming together and nuclei in the center. Now, what happens? No, you take GI tract, suppose example. So, what happens in the GI tract? No, now I'll tell you why the basic tissues are called basic tissues, because they will be forming they are part and parcel of all other organs. The basic tissues, like suppose you take small intestine. Okay, now small intestine is a tubular structure. It is having lumen. So, lumen of it is lined by epithelium. So, below the epithelium, like there is a supporting connective tissue. So, there is a connective tissue. Then after that, what we see is we see muscle layer. So, this muscle layer, which type of muscles are present? Smooth muscles are present. So, smooth muscle. Now, smooth muscles there are arranged in two different pattern. So, longitudinal manner and circular manner. So, in the small intestine there is inner circular and outer longitudinal layer. So, and then outside that again there is a connective tissue, and then in between there is nerves are coming and supplying them. So, one particular organ is having epithelium, having connective tissue, having muscles and having nerves. So, that's why these basic four tissues are forming part and parcel of different organs in human body. So, in today's demonstration we are going to see actually the maybe small intestine or the stomach a slide where we can see this smooth muscle, because skeletal muscle we can take and prepare a slide, but smooth muscle they are very small. So, it's usually we take a one particular organ and we show you the muscle there in that particular organ. So, there also you are going to find their arrangement like circular and longitudinal. So, in longitudinal section we can see that elongated spindle shaped cells, cytoplasm is like eosinophilic dark, and because of the myofilaments which are present, and then nucleus is elongated and lightly stained. Okay. So, and that will be present in the center, and here in the transverse section we see the centrally placed nuclei. Any specific feature we cannot see compared to the Now, if you can see in this particular, this is schematic view, here you can see this is how they are longitudinally arranged, and here in the transverse section how we can see them. So, this is the transverse section and this is the longitudinal section. If you can see here, this, please come. So, here we can see the longitudinal and this is the transverse section. Okay, and this is the other part of that particular organ. So, we will not go in detail which organ is this, but in this we can see this is the circularly arranged muscle, smooth muscle fibers, and these are transverse. This is the transverse section of the smooth muscle fibers. So, in this particular diagram we can see the skeletal muscle, cardiac muscle and the smooth muscle, where you can in one view you can differentiate them. So, see the skeletal muscle fibers running parallel to each other, even cardiac muscle they are running parallel, but in cardiac muscle they branch and again anastomose. Then we can see in the skeletal muscle fiber the nuclei are pushed to the periphery, and each muscle fiber is multinucleated. There are many nuclei, while in the cardiac muscle the nucleus is present in the center. And then in case of smooth muscle, here we can see the spindle shaped smooth muscles coming together, fusing together and forming the smooth muscle, and they are like here, and nucleus is present in the center of it. And yes, we have seen the intercalated disc where the two adjacent muscle fibers in the cardiac muscle they come in contact with each other. Here these are the transverse sections of these three. So, in the transverse section it is difficult to distinguish these three from each other, but in longitudinal section we can easily distinguish skeletal muscle from the cardiac and smooth muscle. We can distinguish them from each other. So we know that this skeletal muscle, their action is strong, right? They quick and they are voluntary in nature. Whereas, you know, the longest muscle fiber is the can go up to the 30 centimeter in length. Do you know which is the longest muscle? Sartorius, right? So it can go up to 30 centimeter in length. While even cardiac muscles are also strong, quick, but they are involuntary in contraction. Their length is not long as compared to the skeletal muscles. And as far as smooth muscles are concerned, they are weak, they are slow, they are involuntary in nature. So that is all about this different type of muscles. So we can see in this this is the transverse section of these three: skeletal, cardiac, and smooth muscle. Okay, and this is the longitudinal section. As I told you, it is easy to distinguish these muscles from each other by the longitudinal section. We can see here they are running parallel. We can see the striations, we can see the nuclei which are pushed towards the periphery, and here can you appreciate this? These are the intercalated discs. So these are basically a high power view. Okay, 40x. You can see this in 40x, and then we can see the centrally placed nuclei. Maybe sometimes two, most of the time one nucleus in the cardiac muscle, and this is the smooth muscle in the transverse section. Smooth muscles are present. So yeah, sorry, longitudinal section. So that is all about the muscles. Any doubt? So who can any volunteer who can differentiate these three, just distinguishing features of these three together? Let us try someone else. If no one volunteering, then we'll come to Maznaz. Anyone? Okay, yes, Maznaz, go ahead.
So the first and the basic differentiating feature is the location of nuclei.
Yeah, go ahead.
In the skeletal muscles, the nuclei are very cardiac, they have a hollow space around the nuclei, so we can differentiate from cardiac and smooth muscles like that.
Yeah.
The skeletal muscles are not branched, they are unbranched and they are parallelly spaced. Other distinguishing feature is being the skeletal muscles, the striations are visible, significantly visible.
Yes.